Topic 1a Molecules and cell components. Q. What is the definition of a molecule? Two or more atoms are covalently bonded together. Examples: H2O, O2, glucose. Large biological molecules include proteins and nucleic acids. Q. What are the 6 especially common elements in cells? Carbon, C, hydrogen, H, oxygen, O, nitrogen, N, phosphorus, P, sulfur, S. Backbones. Functional groups of proteins, nucleic acids, carbohydrates, and lipids. Q. What special properties make water a good solvent? Polarity and hydrogen bonding, ions and polar molecules, hydration shells, high heat capacity, heat of vaporization, thermal stability, cohesion. Adhesion supports transport in organisms. Q. What is an acid? What is a base? Acid, proton, H+, donor. Example: HCl2H++ Cl negative base, proton acceptor or O negative donor. Example: NaO to Na positive plus O negative. Q. What is pH? pH equals negative low plus, measures acidity, alkalinity. pH 7 acidic, pH equals 7 neutral, pH 7 basic. Q. List the 7 categories of molecular and ionic components of cells and their functions. Water, universal solvent. Nucleic acids, store, transmit genetic information. Proteins, enzymes, structure, regulation, transport. Lipids, fats, membranes, energy storage, signaling. Sugars, polysaccharides, energy storage. Glycogen, starch, structure, cellulose. Small organic molecules, metabolic intermediates, cofactors. Inorganic ions, osmotic balance, membrane potentials, enzyme cofactors. Q. What are the 4 main macromolecular components and their building blocks? Proteins, amino acids, peptide bonds. Nucleic acids, nucleotides, base plus sugar plus phosphate. Polysaccharides, monosaccharides, glycosidic bonds. Lipids, fatty acids plus glycerol, not true polymers. Q. Describe D's DNA structure. Double helix with antiparallel strands, 5 to 3 versus 3 to 5. Sugar phosphate backbone, bases inside pair A to T, 2 H bonds, and G to C, 3 H bonds. Major minor grooves enable protein binding. Q. What is the central dogma? DNA to RNA to protein. Replication by DNA polymerase, transcription by RNA polymerase, translation by ribosome. Q. What protein performs DNA replication, transcription, and translation? Replication, DNA polymerase. Transcription, RNA polymerase. Translation, ribosome. RNA plus proteins. Q. Main function of membranes formed by lipids? Selective barriers, phospholipid bilayers, that compartmentalize, mediate transport signaling, and support energy conversion. Topic 1b Bioenergetics. Q. Discuss the overall flow of carbon and oxygen on Earth. Photosynthesis. CO2 is fixed into organic molecules, O2 is released. Respiration. Decomposition. O2 consumed, CO2 released back to the atmosphere. Q. Discuss the overall flow of energy on Earth. Sunlight captured by photosynthesis leads to chemical energy, ATP, NADPH, sugars. Cellular respiration extracts energy from reduced molecules to make ATP. Q. Basic principles of cellular bioenergetics. Catabolism releases energy, electrons, NADH, FADH2, by oxidizing fuels. Anabolism uses ADP, NADPH to build macromolecules, coupling via ATP and reducing equivalence. Q. Energetic molecules, formation and use. ATP, substrate-level phosphorylation and oxidative photophosphorylation, drives endergonic processes. NADH, FADH2, produced in glycolysis, TCA, donate electrons to etc., leading to ATP. NADPH, produced in the pentose phosphate pathway, photosynthesis, used in biosynthesis. Q. Energy storage compounds made by animals versus plants. Animals, glycogen and triacylglycerols, fats. Plants, starch, oils, fats, seeds, storage proteins. Q. CO2 cycle and sun energy fate. CO2 fixed leads to biomass, returned via respiration, decay, combustion. Sun energy drives photosystems. Electrons from H2O reduce NADP+. Energy is stored in sugars. Q. Relation of anabolism and catabolism, oxidized versus reduced. Catabolism, oxidation leads to energy. Anabolism, reduction consumes energy. Oxidized molecules have fewer electrons, less energy. Reduced molecules have more electrons, more energy. Q. Why ATP is the cellular currency, why NADH is currency, not storage. ATP hydrolysis is rapidly available and couplable, regenerated quickly. NADH is a short-term electron carrier for ATP synthesis, not long-term storage. Topic 1c Organelles. Q. Basic features, functions of organelles. nucleus DNA storage, transcription, nucleolus makes RNA, ER, protein synthesis, SER, lipid synthesis, detox, Golgi modify sorts packages macromolecules, mitochondria ATP production, genome, apoptosis roles, lysosomes acidic compartments for macromolecule digestion, peroxisomes oxidation of fatty acids, detox, H2O2, vacuoles, plants, storage, turgor, endosomes, endocytic trafficking, chloroplasts, plants algae, photosynthesis, genome, Q. Prokaryotes versus eukaryotes, origins of mitochondria chloroplasts, prokaryotes, no nucleus organelles, circular DNA, small, eukaryotes, nucleus organelles, linear chromosomes, larger. Endosymbiosis, engulfed bacteria became mitochondria chloroplasts, double membranes, circular DNA. Topic 1D, Molecular Evolution Q. 3 Domains of life, why some genes evolve slowly, relatedness, and genome evolution. Domains, bacteria, archaea, eukarya. Essential genes, e.g., RNA, are constrained, leading to slow evolution. Non-essential regions evolve faster. Relatedness via sequence similarity. Phylogenetics. Mechanisms, mutation, segment shuffling, gene duplication, horizontal transfer. Topic 1E Model organisms Q. Common characteristics, examples, genome sizes, life cycles, traits, short generation time, easy culture, small size, sequenced genome. Examples, E. coli, S. cerevisiae, C. elegans, Drosophila, Arabidopsis, zebrafish, mouse. Genome size range, 4.6 MB, E. coli, to 2.8 GB, mouse. Life cycle range, minutes, E. coli, to months, mouse. Topic 2 Proteins and enzymes Q. Chemical nature of amino acids. Proteins, important aspects of protein structure. Amino acids, central C with amino, carboxyl, H, and side chain, R. Zwitterions at physiological pH, multimers bonds. Protein structure, primary, sequence, secondary, A helix B sheet, tertiary, 3D fold, quaternary stabilized by hydrophobic effect, H bonds, ionic interactions, van der Waals, and disulfide Q. Why are proteins more diverse than DNA? Side chain categories, 20 chemically distinct side chains versus 4 nucleotide bases. Side chains, acidic, basic, polar uncharged, nonpolar hydrophobic. Q. What is a protein domain? How does the DNA sequence relate to domains? Domain, discrete structural functional unit in a protein, e.g., kinase domain, encoded by consecutive stretches of DNA. Domains can be modular reused. Q. What is a ligand? What kinds of molecules can be ligands? Ligand, any molecule ion that binds specifically and reversibly to a protein, or nucleic acid. Can be ions, Ca2+, Zn2+, small molecules, glucose, CAM, lipids, nucleotides, ATP, peptides, proteins, DNA, RNA segments, even water. Binding often occurs at a defined site, active site, allosteric site, receptor pocket. Q. What determines protein binding to a ligand? Structural complementarity, shape fit, and chemical complementarity, H bonds, ionic, hydrophobic, van der Waals. Dynamic induced fit, protein ligand adjust conformation upon binding. Environmental conditions, pH, ionic strength, temperature, presence of cofactors or metal ions. Q. Does noncovalent binding imply weak binding? Individual noncovalent interactions are weak, but many together can produce a high affinity, tight binding. Affinity quantified by Kd, dissociation constant, lower Kd equals stronger binding. Q. How do enzymes catalyze reactions? Lower activation energy by stabilizing the transition state. Orient substrates, exclude water, provide catalytic residues, acid-base, covalent catalysis, metal-ion catalysis. Induced fit optimizes interactions at the active site. Q. What is the Michaelis-Menten equation? V equals Vmax, S, Km plus, S. Describes the initial reaction velocity versus substrate concentration for many enzymes. Q. What is Km? Units? Km equals S at half Vmax. Lower Km indicates higher apparent affinity. Units, concentration, e.g., m, mm, mm. Q. What is Vmax? Units? Vmax equals maximal rate when the enzyme is saturated with substrate. Units, amount per time, e.g., mol, Sn to the power of 1, or normalized per enzyme amount. Q. If Km is large, is the enzyme good? Large Km implies lower apparent affinity, more substrate needed for half Vmax. Good depends on context, substrate availability, physiology. Q. Do enzymes catalyze catabolic or anabolic reactions? Both. catabolic, breakdown, energy release, and anabolic, biosynthesis, energy consumption. Q. What is a metabolic pathway? How are pathways regulated? An ordered series of enzyme-catalyzed reactions converting substrates to products. Regulation, feedback inhibition, allosteric activators inhibitors, covalent modification, phosphorylation, transcriptional control, compartmentalization. Q. What is protein phosphorylation? How can it alter activity? Covalent addition of phosphate, by kinases, to CR-THR-TIR, removed by phosphatases, introduces a negative charge leading to conformational change, can activate and inactivate enzymes, alter localization, or modulate interactions. Topic 3 DNA, replication and chromatin. Q. Flow of genetic information, central dogma, and key enzymes complexes. Replication, DNA polymerase copies DNA, requires primer, 5-3 synthesis, proofreading. Transcription, RNA polymerase synthesizes RNA from the DNA template. Translation, ribosome decodes mRNA into a polypeptide with tRNAs. Q. What is a gene? A DNA sequence that encodes a functional product, protein or functional RNA, includes coding regions and regulatory elements. Q. DNA structure recap. What does antiparallel mean? What does semi-conservative mean? Antiparallel, the two strands run in opposite 5-3 directions. Semi-conservative, each daughter DNA duplex contains one parental and one new strand. Q. Replication details, including chromosome ends. The leading strand is synthesized continuously, the lagging strand is synthesized discontinuously as Okazaki fragments. RNA primers are removed, and gaps are sealed by DNA polymerase DNA ligase. Linear chromosome ends are replicated by telomerase, which extends telomeres using an RNA template. Q. DNA repair mechanisms. Proofreading, 3-5 exonuclease, by replicative DNA polymerases. Mismatch repair, post-replication, corrects mispaired bases. Base excision repair fixes small base lesions, e.g., deamination. Nucleotide excision repair removes bulky lesions, e.g., UV dimers. Double-strand break repair, non-homologous end joining, NHEJ, or homologous recombination HR. Q. What is a chromosome? What is chromatin? Basic unit of chromatin? Chromosome, a DNA molecule with associated proteins carrying genetic information. Chromatin, DNA plus histones plus non-histone proteins. Nucleosome, 147 BP DNA wrapped around histone octamer, 2XH2A, H2B, H3, H4. Q. Euchromatin versus heterochromatin, packed versus unpacked chromatin. Euchromatin, less condensed, transcriptionally active. Heterochromatin, highly condensed, transcriptionally silent, e.g., centromeres, telomeres. Packed equals inaccessible, unpacked equals accessible to transcription factors. Q. What is a histone tail? What does it mean that chromatin is dynamic? Histone tails, flexible N-terminal regions protruding from nucleosomes. Dynamic chromatin, post-translational modifications and remodeling change compaction accessibility over time. Q. What is a remodeling complex? Histone modifications and their significance. Chromatin remodeling complexes reposition evict nucleosomes using ATP. Histone modifications, acetylation, methylation, phosphorylation, ubiquitination, form a histone code influencing gene expression, DNA repair, and replication. Q. Example of compact chromatin. What is a code reader complex? Compact example, paracentromeric heterochromatin. Code reader, protein domain, e.g., bromodomain, chromodomain, that recognizes specific histone marks to recruit effector complexes. Q. How does chromatin condense with remodeling complexes? Reader-remodeler cooperation spreads marks and repositions nucleosomes to build higher-order compaction, e.g., during mitosis. Q. What do DNA and RNA stand for? Building blocks of DNA versus RNA. DNA, deoxyribonucleic acid, RNA, ribonucleic acid. DNA building block, deoxyribonucleotide, deoxyribose plus base plus phosphate. RNA building block, ribonucleotide, ribose plus base plus phosphate. U replaces T. Q. Base pairing rules. What does antiparallel semi-conservative mean? Recap. DNA, AT, 2H bonds, GC, 3H bonds, RNA, AU, GC. Antiparallel, opposite strand orientations, semi-conservative, one old plus one new strand after replication. Q. Problem with lagging strand. Outline the replication process. Name 6 proteins and functions. How do eukaryotes speed up replication? What is the replisome? Lagging strand is synthesized as Okazaki fragments due to 5-3 polymerase directionality. Replication steps, origin firing leading to helicase unwinds, leading to primase lays primers, leading to DNA pole extends, leading to SSB stabilizes ssDNA, leading to ligase seals NICs, leading to topoisomerase that relieves supercoiling. 6 proteins, helicase, primase, DNA polymerase, SSB, DNA ligase, and topoisomerase. Eukaryotes speed replication with many origins firing per chromosome. Replisome, multi-protein machine coordinating unwinding and synthesis at forks. Drawing a DNA diagram, 2 rows of sugar phosphate backbones on top and bottom. Top left of backbone is 5 and bottom left of backbone is 3. Top right of backbone is 3 and bottom right is 5. This makes them anti-parallel. Nucleotides starting from top left is alpha.